Good morning, dear students. Today, let's discuss about the structure of plant cell. Students, have you ever wondered how a silent plant standing in one place can manufacture food, respond to stress, store water, communicate chemically, and even defend itself against pathogens? All these extraordinary functions begin with one fundamental unit, that is the plant cell. As the proverb says, a building is only as strong as its foundation. Likewise, the strength of plant, physiology, growth, metabolism, and adaptation lies in the remarkable organization of the plant cell. Before we begin, let me ask you a question. If a plant cell and an animal cell were placed side by side under a microscope, what are the first differences you would identify? Cell wall, chloroplast, large vacuole, shape differences. Excellent. Those differences are not merely structural, they define the lifestyle of plants. A plant is autotrophic, sessile, and continuously exposed to environmental stress. Therefore, every organelle inside the plant cell has evolved with specific adaptations. Let me define plant cell. A plant cell is the structural, functional, physiological, and hereditary unit of plants. It is a eukaryotic cell characterized by a true nucleus, membrane-bound organelles, cellulose cell wall, plastids, large central vacuole. Most plant cells range from 10 to 100 micrometers in size, although specialized cells may vary considerably. Now, let us briefly travel back to the history. In 1665, Robert Hooke observed thin slices of cork under a microscope and coined the term cell because the compartments resembled monastery rooms. However, Hooke actually observed dead cell walls, not living protoplasm. Later, Matthias Schleiden and Theodor Schwann proposed the cell theory. And finally, Rudolf Virchow stated that every cell arises from a pre-existing cell. Even today, this principle remains the fundamental in biology. Let me now explain the overview of plant cell structure. Structurally, the plant cell may be divided into three major regions: cell wall, protoplast, and cell inclusion. The protoplast includes the plasma membrane, cytoplasm, nucleus, and all organelles. First, let us see the cell wall. It is the outermost non-living rigid layer surrounding the plasma membrane. But do not underestimate it. Although non-living, it plays a dynamic role in protection, communication, transport, and mechanical support. As the saying goes, discipline gives strength. Similarly, the cell wall gives structural discipline to the plant cells. The primary component of the plant cell is cellulose, a linear polymer of beta-1,4 linked glucose units. The other components include hemicellulose, pectin, lignin, suberin, cutin. Depending on the cell type and function, the wall composition may differ. There are three layers in the cell wall. First is middle lamella. It is rich in calcium pectate and acts as a cementing layer. The second is primary cell wall, which is flexible and extensible. It is found in actively growing cells. Third is secondary cell wall. It is thick, lignified, and rigid, which is seen in mature cells like xylem vessels. Students, now think critically. Why do xylem cells require thick secondary walls? Anyone knows? No answer. Okay, actually, to withstand negative pressure during water transport. One of the fascinating features of plant cell is plasmodesmata. These are microscopic cytoplasmic channels connecting the adjacent cells. They allow symplastic transport, cell-to-cell signaling, RNA and protein movement. So, plant cells are not isolated compartments, they function as a coordinated community. In modern plant biology, plasmodesmata are considered as communication highways. Next is plasma membrane. Just beneath the cell wall lies the plasma membrane. According to the fluid mosaic model proposed by Singer and Nicholson, the membrane consists of phospholipid bilayer, proteins, sterols, carbohydrates. The membrane is selectively permeable. Here is a question for you. If the plasma membrane loses selective permeability, what will happen to cellular hemostasis? The cell cannot regulate ion exchange, water balance, or metabolic transport efficiently. Next is cytoplasm. The cytoplasm is the semi-fluid matrix between the plasma membrane and the nucleus. It consists of cytosol, organelle, cytoskeleton, and metabolic enzymes. Numerous biochemical reactions occur here, including glycolysis and intermediary metabolism. Let me say something about nucleus. The nucleus is often called as control center of the cell. However, modern molecular biology views it as the genetic information hub. The nucleus is surrounded by a double membrane known as nuclear envelope. The nucleus contains nuclear envelope, nucleoplasm, chromatin, and nucleolus. The nucleolus will synthesize ribosomal RNA. Chromatin consists of DNA associated with histone proteins. During the cell division, the chromatin condenses into chromosomes. Please respond, students. Why is the nucleus absent in mature sieve tube elements? To maximize the efficiency in translocation of photosynthesis, companion cells also support sieve elements metabolically. Coming into endoplasmic reticulum, it forms an interconnected membrane network. Two types are present. One is rough endoplasmic reticulum that contains ribosomes involved in protein synthesis, and the other is smooth endoplasmic reticulum that lacks ribosome but it functions in lipid synthesis and detoxification. Think of endoplasmic reticulum as the intracellular transport and manufacturing system. Coming into ribosomes, these are the site of protein synthesis. Plant possess 80S ribosomes in the cytoplasm and 70S ribosomes in mitochondria and chloroplast. This observation strongly supports the endosymbiotic theory. Next is Golgi apparatus. In plants, Golgi bodies are also called as dictyosomes. The functions include packaging, secretion, cell wall polysaccharide synthesis, and vesicle formation. Without Golgi body secretion, cell wall assembly would be severely affected. Now let me say about mitochondria. Mitochondria are known as the powerhouse of the cell. They are double-membraned organelle containing their own DNA and ribosomes. The main function is ATP production through aerobic respiration. Here is the thought-provoking question. Why do actively growing meristematic cells contain numerous mitochondria? Because growth demands enormous energy. Plastids are unique to plant cells and some protists. They arise from proplastids. Three major types are present: chloroplast, chromoplast, and leucoplast. Chloroplast are the centers of photosynthesis that contains outer membrane, inner membrane, stroma, thylakoids, and grana. The green pigment chlorophyll is embedded in thylakoid membrane. Girls, remember this proverb: where there is light, there is life. In biological terms, chloroplast convert the light energy into chemical energy. Can photosynthesis occur without chloroplast? In higher plants, no. But interestingly, photosynthetic bacteria perform photosynthesis without true chloroplast. This again supports the evolutionary adaptation. Now, know about chromoplast and leucoplast. Chromoplasts contain carotenoid pigments and provide color to flowers and fruits. Leucoplasts are colorless storage plastids. The types include amyloplast for starch storage, Elaioplast for oil storage, and Proteinoplast for protein storage. Now coming into vacuole, one of the most prominent structure in mature plant cell is the central vacuole. It is surrounded by a membrane called tonoplast. The functions include storage, maintenance of turgor pressure, pH regulation, waste sequestration, and pigment accumulation. A large vacuole allows the plant to maintain rigidity without expending excessive energy. This is biological efficiency. Coming into cytoskeleton, plant cells also possess an internal framework called cytoskeleton. The components are microtubules, microfilaments, and intermediate filaments. The functions include organelle movement, cell division, intracellular transport, and cell plate formation. Without the cytoskeleton, cellular organization will be collapsed. Plant cell contains specialized microbodies such as peroxisomes that involved in photorespiration and detoxification of hydrogen peroxide. Glyoxysomes that convert stored lipids into carbohydrates during the seed germination. These organelles are especially important in oil-rich seeds. Plant cells also accumulate ergastic substances or cell inclusions. The example includes starch grains, aleurone grains, crystals, tannins, and oils. And these are usually non-living deposits with storage or protective functions. Thank you girls for your patient listening.